Good morning and welcome. It's 8 o'clock, so we will make a prompt start. It's wonderful to see so many of you here this morning at the final day of what has been an absolutely wonderful Congress at WPC 2023. Welcome to this session on hot topics. My name's Emily Henderson. I'm an Associate Professor of Ageing and Movement Disorders at the University of Bristol in the UK. And it's my great pleasure to chair the session this morning. We have 4 absolutely superb speakers talking about the very latest in developments in Parkinson's disease. So without further ado, we'll move on to the first of our 4 talks this morning. So I'm delighted to welcome Dr. Christin Weissleder, originally from Germany but now based in France, studying the etiology of Parkinson's. Dr. Weissleder, welcome.
Okay, thanks Emily for the introduction, and thanks to the organizing committee for giving me the chance today to show my work. I'm a postdoctoral fellow in the group of Michela Delighty at the Imaging Institute in Paris, where we try to understand the role of the gut-brain axis in the pathogenesis of Parkinson's disease in patients that are carrying the GBA mutation. I have nothing to disclose. So although gastrointestinal dysfunction has already been described by James Parkinson 200 years ago, it was only in the last 2 decades that research is really focused on the role of the gut-brain axis in the onset and progression of Parkinson's disease. When we look at the patients, what we do find is that the microbiome has changed, and one of the most consistent findings is that we have a reduction in short-chain fatty acids. These changes have been linked to changes in the epithelial barrier maintenance as well as inflammation. And when we look at the tissue from patients with Parkinson's disease, we do see alpha-synuclein aggregation. And that has been speculated to be then transmitted via the enteric nervous system to the brain. Other changes in the brain also include, again, inflammation and the activation of brain-resident immune cells, the microglia. And then we do see the loss of dopaminergic neurons in the substantia nigra later in the disease stage that then lead to the onset of motor symptoms. This entire gut-brain axis is not unidirectional, so it can also go the opposite way. And what has been quite interesting that we can, of course, target changes in the gut with microbial targeted therapies like probiotics, prebiotics, and other diet modifications. That would be a very easy way to intervene at a very early disease stage. As I mentioned, we do look at patients that are carrying the GBA genetic risk. So GBA encodes the lysosomal enzyme glucocerebrosidase that catalyzes the hydrolysis of glucosylceramide to glucosinceramide. What is interesting is that only 30% of the patients that do carry the heterozygote mutations in GBA develop Parkinson's disease. And from the clinical perspective, these patients do show very often gastrointestinal dysfunctions. As we already heard during the conference, they have a faster disease progression and an earlier disease onset. When we look at the animal models, there have been some studies that try to restore GCase activity, and in the gut, we can rescue the pathology and function of this phenotype. This then led to the question of whether we have an interaction between GBA and microbial changes. And in our group, we're using iPS-derived in vitro model systems. And so far, we have received fibroblasts from the patients with Parkinson's disease carrying different GBA mutations. And we have performed the CRISPR gene correction to have the corresponding gene-corrected control. And we've also used control cells, since there is a lot of controversy about the role of these microbial factors in inflammation and in general in gut function. For the gut part, we then differentiated these cells into macrophages as the peripheral immune cells, into peripheral neurons as the link between the gut and the brain, into intestinal organoids, so kind of mini gut system, as well as more complex organoids where we combine immune cells and peripheral neurons with the intestine. For the brain perspective, we have been generating microglia as the resident immune cells in the brain, as well as midbrain organoids that was recently published. And are currently in the process of also combining these two populations to investigate the role of the gut or microbial factors in this setting. For the GBA lens, we have performed a histological, biochemical, and functional characterization. And today, I really would like to focus on the macrophages and the microglia, as well as show a few findings on the intestinal organoids. So for the macrophages, we have been characterizing the enzyme activity, the expression of GBA, as well as the ability to secrete cytokines, as well as perform phagocytosis, so the clearance of debris. You can see here in the pictures that when you carry the more mild mutation, the E326K mutation, that the macrophages differentiate quite nicely and express the markers that we would expect. We do see a reduction in GCase activity, or with a mild phenotype, while the GBA expression remains unchanged. For the phagocytosis, we can see that there's a reduction in the capacity of these immune cells to take up beads that we're adding to the cell culture. And for the cytokine secretion, what you can see here in black is always the gene-corrected, what we refer as wild type, as well as the E326K mutation in red. And when we add LPS as a bacterial endotoxin, we can see that the capacity of these immune cells to respond to LPS is impaired. We have done the same experiments for the microglia, the brain resident immune cells, and what we can see here again is that they differentiate quite nicely into microglia expressing the specific markers PU1 and IBA1. We don't see a significant change in GCase activity, and also the GBA expression remains unchanged. But what was very interesting, and similar to what we have found with the macrophages, is that the ability to phagocytose is impaired in these immune cells that are carrying the mutation, and similar to the cytokine findings with the macrophages, we also see the same pattern of a reduced secretion of pro-inflammatory cytokines in the mutant. We have done the same experiments with a more severe mutation, which is the L444P mutation, where we have a heterozygote and a homozygote, so you can see a very nice stepwise reduction in the GBA expression. That was also reflected in the GCase activity for the macrophages. And here, just as an example, we again see a reduction in the secretion of pro-inflammatory cytokines like TNF-alpha. We had a very similar pictures for the microglia. Again, the patterns are very similar in the reduction in GBA expression, GCase activity, and cytokine secretion. Then for this phenotype, what was very interesting is that when we checked for other chemokines, so chemokinatic cytokines, we found two that stood out to us, and these were CXCL10 and CCL17, because they did show an opposite pattern. So when we look at the macrophages, the secretion is reduced upon stimulation, and for the microglia, the secretion is increased. That led to the question, like, what are these chemokines doing? So when we look at CCL17, it's also known as TARC, or thymus and activation-regulated chemokine. The receptors are expressed by T cells, so it is involved in the recruitment of T cells and other peripheral immune cells. And a very similar function, but more focused on another type of T cells, is known for CXCL10, which is also known as the interferon gamma-inducible protein. But again, it is involved in the recruitment of T cells, which then led for us to kind of look at, like, are these immune cells recruiting, or like, is there a system to investigate whether there's immune cell recruitment in our system? So we're using a brain or intestine on chip model, and here you can see that we have been successfully generating intestinal and midbrain organoids, and we use the system where we can see these intestinal organoids in the top channel, and in the bottom channel we are seeding endothelial cells, and then this allows us to really investigate whether there's increased immune cell recruitment in the system. So this is work that we're planning to do very soon. Then for the organoids, we have so far been characterizing the different cell populations for the lines with the GBA mutation. And here you can see that we do find the classic populations that we would see also in the human gut, like the enteroendocrine cells that are secreting the neurotransmitters. We do see the goblet cells that are producing mucus. For the mild mutation, we already see in these organoids at day 30 of differentiation that there's a reduction in GCase activity, and we also see a reduction in GBA expression that was then associated with an increase in alpha-synuclein expression. And this was also seen with this kind of correlation indicating that the enzyme activity that is involved in the turnover of alpha-synuclein, once it's reduced, you do accumulate this protein. To sum up where we're at the moment in trying to understand the interaction between GBA and microbial factors, so far I've shown you that the cytokine secretion upon stimulation is impaired in immune cells that are carrying the genetic risk, and they also show impaired phagocytosis. We do see this very interesting finding of increased CXCL10 and CCL7 secretion by microglia that are carrying the L444P mutation that points towards genotype-specific differences in the inflammatory response. For the organoids, we have the reduction in the GBA expression that is associated with increased alpha-synuclein expression. And we would like to now characterize using this organ-on-a-chip model to see whether the role of GBA and microbial factors in inflammation in the system. And last but not least, what is also always very important for us to see whether we can validate our findings in patients with the disease. So we have been receiving blood and cerebrospinal fluid, as well as post-mortem tissue from patients with the GBA mutations, where we would like to do validation studies. With that, I would like to thank Michaela, as well as everyone else from the team who has been helping with the project. Our collaborators, funding sources, and of course the patients who are always participating, as well as you for coming today. Thank you.
Dr. Weissleder, thank you so much. What a fantastic and stimulating talk to start this morning. Just a reminder, please, in the audience, we can take questions from the floor. If you don't want to ask a question using the microphone, you can use the app. And we'll take questions after each talk, and then if there's some time at the end, we'll use that for questions as well. So any questions from the floor for Dr. Weissleder? Okay, and nothing on that app. Christin, I wondered if you could just comment about what you think might be the role of other microbial factors.
So yeah, when we look at the patients with Parkinson's disease, one of the most consistent one is the changes in the short-chain fatty acids like butyrate, acetate, and propionate. So what we have done so far is, since there's still quite a bit of controversy regarding the role of these short-chain fatty acids in general, we have been treating control cells without the mutation with these short-chain fatty acids. And what we found is that when we treat them just with a single short-chain fatty acids like butyrate, we rather have a pro-inflammatory profile of these immune cells for macrophages and microglia. And we have also analyzed other changes beyond inflammation that indicate that these short-chain fatty acids could have a role in increasing lipid storage, which is of interest for these diseases, so yes. And then we would translate it to apply and treat the cells with the GBA mutations.
Thank you very much indeed. Any other questions from the floor? Lady over here.
I have a question. So all the studies time and time again show that we tend to be high in bifidobacteria and lactobacillus in the studies, the microbiome studies. Have you any view on that? Because they're supposed to be anti-inflammatory, but you know, why they're so high in people with Parkinson's?
We haven't looked at, so we have rather focused on the changes that are decreased in Parkinson's. And one of the most common ones, as I mentioned, are the changes in short-chain fatty acids. So that was our primary focus. Of course, it's very complex. So it's not just the ones that are reducing, also the ones that are increasing, like what you mentioned with lactobacillus. So yes, I think one option would be that we use our system to really add the changes that we see in the patients to our system with the microbiome and investigate in a more complex setting how that could impact pathology.
And do you think gender could have any impact, or in terms of sex, have any impact? Have you seen any signals in terms of changes there, just out of interest?
Our lines are all deriving from male patients so far, so that's what I can say. Of course, there's always a gender effect in Parkinson's disease, but I think for the short-chain fatty acids, I'm not aware of any studies that have identified more gender-specific things.
Okay. Maybe we'll see some in the future.
Yes, that would be great. Thank you.
Perfect. Lots of food for thought and potential future avenues. Thank you.